Friends, today we are going to discuss about one of the important topic in developmental biology about pattern formation in Drosophila melanogaster. No? Pattern formation in Drosophila melanogaster. Drosophila melanogaster is along anterior posterior axis. In previous classes we have discussed about blastulation and gastrulation. Gastrulation ensures that the three primordial germ layers, ectoderm, mesoderm and endoderm, should be properly placed through varied morphogenetic movements. We have discussed at length. And what next after gastrulation? After gastrulation, there should be organogenesis. And for organogenesis, there should be a proper pattern. What exactly that pattern? What is a pattern formation? Pattern formation is nothing but acquiring of different cellular identities depending on the spatial arrangement in a developing embryo. That means, say for example, a group of cells will differentiate and organize to form head, and a group of cells to form a tail. That means depending on different spatial arrangement. This spatial arrangement is most important. At the anterior end, it will form head. At the posterior end, it forms tail. Likewise, that means this acquiring of different cellular identities is the crux point for proper organogenesis. So this pattern formation is like fundamental block for for the smooth conduct or process of organogenesis. Okay, here advancements in the forward genetics concept, particularly in the late, that means in the aftermath of 1990s, broaden the concept of pattern formation. That means what exactly the forward genetics aids in pattern formation? It gives us the idea about different genes responsible for the pattern formation. Say how they have conducted, that means the scientists conducted different mutagenetic experiments. So for example, if X gene is responsible for a particular structural pattern, they have subjected the same to a radiation so that they can identify which group of genes, or that means gene clusters, are responsible for this pattern formation. So therefore here Drosophila melanogaster is considered as a favorite object for scientists. Why? Because it is hardy, it can be adapted to diversified conditions, and it is abundant. And actually it is nothing but a tiny dipteran insect, say it around 3 millimeters. The adult insect is just around 3 millimeters, and it can be easily available. It is commonly known as fruit fly. Okay, today we are going to discuss about this concept. Okay, and regarding the basic issues, we have discussed now genes responsible for pattern formation. We have to identify the first important. Here the pattern formation broadly involves two concepts. The polarity, that means what we call polarization, and segmentation. Here there are different genes responsible for polarization and segmentation. Polarity is by means of maternal effect genes. Maternal effect genes. Segmentation is by means of zygotic genes. Before we go into the main topic, we have to discuss some basics of Drosophila. Drosophila melanogaster, we have discussed a tiny animal. Here the thing is the oogonia. We have discussed in different issues of oogenesis. Here it doesn't undergo complete divisions. 4 karyokinesis but incomplete cytokinesis. That leads to formation of 16 cells which are interconnected, out of which 15 cells serve as nurse cell and 1 cell is a oocyte. And these nurse cells, they contain maternal effect genes. Here all these are before the fertilization. This is most important. That means polarization, which brought down by maternal effect genes, will brought down before the fertilization process, and they translate different mRNAs, and these mRNA they wait until fertilization, and they are spatially distributed across the different regions of egg. This is the basic point, the crux point for polarity. Different mRNA concentrations. In the long run, they form different, that means they translate into proteins, and their protein concentrate different, what we call concentration gradient of proteins, which enables us to identify different poles. This is an anterior, posterior. We have discussed already. Along the anterior posterior axis. That means polarization should be along, that means a particular manner. How? See this by means of maternal effect genes. Say for example, this is the rough sketch I am drawing. This is the oocyte, this is the glowing egg. Then only one cell will be serving as oocyte. The rest will be considered as nut cells. That means 15 cells totally, 15 cells totally. 15 cells. The nut cells, and these are the oocyte, and these are the follicle cells. Later on, what we will discuss in the previous classes, centrolethal eggs, the type of egg present in insects is centrolethal. That means yolk is distributed in the center, whereas the syncytial nuclei, repeated karyokinesis without cytokinesis, up to 12th nuclear divisions. Later onwards, after the 13th nuclear divisions, the cell membrane invaginates to form different cells. Here we can consider it as syncytial blastrodom. After fertilization, it becomes, for example, it is actually a sausage-shaped egg. Then out of which the cells, the periphery of the cells, will be occupied by the cells. Syncytial blastrodom, what we call. In the center, there will be yolk. There will be yolk. The centrolethal eggs actually, centrolethal eggs. What happens? We have discussed already. Polarization starts by first the maternal effect. Then they synthesize, that means they translate into different mRNAs. Say the nut cells, and these mRNAs through interconnection, actually they are connected. These cells, they are not completely detached. They are connected to interconnections through microtubules. They reach the oocytes, mRNA. Then after fertilization, we have discussed already, the cleavage will start, and these mRNA, they will translate into proteins, and these proteins, what we call maternal proteins, will diffuse into syncytial blastrodom. Will diffuse. That means here these proteins which form concentration gradients, so that we can identify different poles, anterior and posterior poles likewise. So that this is that leads to polarization, anterior and posterior along the anterior posterior axis. So that further segmentation will be formed. We already discussed pattern formation will be two steps: polarity formation by maternal effect gene, then the segmentation. After the zygote formation, the zygotic genes will take care of it. Okay, this is about the first step. And what are the maternal effect genes? Then they enter in the different blastrodom cells, syncytial blastrodom, and undertake the next process. Here, what are the maternal effect genes that are responsible for this? Bicoid, nano, then torso. 3 classes of maternal effect genes. 1 bicoid genes, 2 nanos, and 3 torso. Here, bicoid genes are responsible for anterior part. Let me say anterior end. They will take care of the anterior end. Nanos, the posterior. Torso, the extremities of anterior and posterior. Say, in the way of acron and telson. The point is simple. By the maternal effect genes translate to mRNA, then the proteins, and these proteins after they diffuse, and they will take care of different regions of anterior posterior axis. Here bicoid will take care of anterior part, the nanos in the posterior part, then the torso, the extremity, that means the acron at the anterior, telson at the posterior. We already discussed. Cells have to acquire different identities based on their spatial arrangement. Anterior, posterior, and extremities. And these genes aids in that process. That means pattern formation, first polarity, then and this leads to zygotic genes. That means how the maternal effect genes, mRNA and protein, and these proteins which are nothing but the transcription factors. Say along with that we have bicoid. It also considered as anterior morphogen. Morphogen means proper structural pattern. That means at the anterior end is taken by bicoid proteins. And these maternal effect protein, by what we call simply maternal proteins, will activate the zygotic genes. That's the connection. The maternal genes are the bicoid, and annos and torso. Okay, here, but and these they activate the zygotic genes. What we call in a cascading manner. And these zygotic genes in a sequential manner they form segmentation. Segmentation. We knew that the basic body plan of insect. It's about basic head, thorax, abdomen. A distinct head. Then a continuous repetitive sequence. That means in the form of repetitive segments. Then the distinct tail. Distinct head, segments, different tail. And this segmentation, what we call this segmentation pattern along the anterior posterior axis done by the maternal effect genes will be performed by the zygotic genes. That's the connection. And how they perform? These what we call transcription factors. They serve as, that means the maternal protein, they serve as transcription factors. Transcription factor which activates like gene on. Gene expression is on. Or gene expression is off. Transcription factor. They activate the zygotic genes. And what are those zygotic genes? Say the gap genes. The gap genes. The pay rule gene. The segment polarity genes. Homeotic selector genes. The gap genes, the pay rule gene, the segment polarity genes, and the homeotic polarity genes. Selector genes. They're all zygotic genes. It's like they activate this. First, for example, bicoid. They activate gap genes, and the gap genes activate the pay rule genes, and the pay rule activate the segment polarity genes. That means in a cascading, we told already, X activate Y, Y activate Z, like that. These maternal effect proteins, they activate the zygotic gene. Say the first zygotic gene to get transcribed under the influence of the activation of maternal effect genes is gap genes. Then it activates the pay rule genes. It activates the segment polarity genes. This is how the action. It is about the basics. Okay, we'll discuss one by one. So it's about the polarity. Therefore, we'll discuss about polarity, how the polarity is formed. Then the zygotic. The zygotic pattern is most important. No, gap genes. Say we have discussed already different genes responsible for segmentation and also the pattern formation. Say the zygotic genes. Say the first gap genes. Hunchback genes. Krupel genes. Giant. Bottom head genes. These are really important. And they are influenced by the maternal effect genes, activated by what we call maternal effect genes. They activate this. Hunchback genes, the Krupel genes, the giant genes, and the butthead genes. These are different gap genes. What exactly the gap? Mutation in the gap gene leads to formation of gaps in the segmentation process. That's it. Formation of gaps in the segmentation pattern is meaning of gap genes, and these are very important. They actually they can define as differences in the region. Differences in the region along the anterior posterior axis that leads to formation different. That means it enables the proper segmentation pattern through activity of different genes. Say hunchback genes, particularly for example, say how I'm just giving it a representation. Say these are the hunchback genes. Hunchback genes. They determine the anterior part. Hunchback. And the cripple. Then the cripple. Then from the center, I don't know, different regions, you know. Then the giants. The giants. Giant genes, then the cripple genes. That means the center. Here the anterior. You know, what exactly how they role perform, we have all discussed now. Maternal effect genes activate the gap genes, say bicoid genes. The bicoid genes, the mRNA, then the bicoid protein will activate the hunchback genes. That means hunchback gene means, say for example, say this is the gene bearing portion. So this particular part is hunchback genes. Then these are the bicoids. That means DNA binding domain, no, for example. And this says mRNA, then the protein. That's it. That means transcription factors. That means they regulate the transcription. Then these hunchback genes, which also serve as a morphogen, It's like the bicoid, the anterior morphogen, the same concept here. The anterior morphogen here, it also serves. That means apart from transcription factor, it also acts as a morphogen. And the rest of the gap genes depends on the hunchback genes, because they are the morphogen, and the rest of the gap genes, they respond to the hunchback genes. And here how they respond, for example, I will tell you a simple example. Here, say the cripple genes. That in order to activate the cripple genes, there should be low hunchback genes. That means, say there are different factors. Higher bicoid and lower hunchback is essential for cripple genes. Otherwise, that means say higher hunchback gene, that means their protein in the sense activity, okay, in the sense that leads to lower activity of cripple. That is why they restrict to center. The most important question, why they restrict to center but not a particular whole area like the hunchback? Because here it acts as a negative regulator. That means the hunchback proteins will not allow the cripple genes to activate if their level is above a particular threshold level. We'll discuss how it ends. This is very important. I told you now. So here the rest of the genes respond to hunchback genes. So somehow the different regions will be formed based on these gap genes. The hunchback genes, cripple gene, but there are other genes are also there. So these are hunchback, cripple, and giant genes. So as we have discussed how these hunchback genes will influence. For example, take a cripple gene activity. Cripple gene in wild type versus mutant bicoid. Okay, the point is bicoid is essential for hunchback, and any variation in the bicoid leads to low production of hunchback, so that allowing the filter, that's a variation, that's the correction. For example, wild type, in general we expect a normal, let me restrict to the center. Say if you try a graph, the hunchback protein, okay, there will be higher hunchback proteins, and say it is a threshold level. So up to this is called activation, later on repression. Here higher level, so that's why they restrict to cripple gene, restrict to center. Say another mutant, so there is no bicoid, so there is no hunchback, low level. Say like this. Here you can expect the cripple protein activity up in the anterior part. Why? Because here the graph represents, say this is the threshold level and low level of hunchback protein. This is the threshold level, low hunchback protein. That's why you can expect the cripple in anterior part, whereas in normal wild type just restrict to the center because of higher hunchback activity. Then here higher bicoid that leads to higher hunchback. But higher bicoid and lower hunchback activity is essential for cripple protein. That means for comparison we have drawn this. Okay, that's how that means these zygotic genes for that means the gap genes they react. The second most important gap zygotic genes here are pay rule genes. Pay rule genes. Pay rule genes. Pay rule genes. Say for example Haley genes and paired genes. Say what exactly the paired genes? Say for example this is the developing embryo. They form striped 7 transverse bands. 7 transverse striped bands, what we call. That means perpendicular to the anterior posterior axis, the pay rule genes, that means the mRNA and proteins, the genes implied means the mRNA and the subsequent translated proteins are responsible for the formation of 7 transverse bands perpendicular to the anterior posterior axis. This is the first step for periodic formation. Then the pay rule genes, that means the pay rule proteins, they activate the segment genes. Segment polarity genes in a cascading fashion, and these segment polarity genes will divide the embryo into 14 segments, what we call periodicity, establishment of periodicity. In the long run, that leads to organogenesis, what we call division of labor. Say for the anterior posterior axis, what we call polarization, then first the differential gradation, say what we call gap gene activity, different gene responses, then the pay rule genes, that means formation of stripes, what we call 7 transverse bands, then the segment polarity genes, they form for 14 segments, then the homeotic selector genes, which determine the fate of each segment. Developmental fate of each segment will be determined by the homeotic selector genes. That's the sequence. That means these are all interdependent actually. Say the pay rule genes should get, that means their activity, their gene expression is depending on that means different concentrations and combinations of gene gap genes. And these pay rule genes, they activates the selector genes, segmentation gene. That's the sequence in a cascading manner. And these are the pay rule genes. They form segmentation pattern. Then there are, for example, Haley genes and paired genes. After that, we have discussed already segment. The third, zygotic genes, segment polarity genes. Segment polarity genes. Discuss already, it's a rough sketch, something like this. It form different segments. Different segments. It will form 14 segments. That means periodicity establishment. So the segment polarity genes. So the fused genes, for example, the patched genes. I'm giving few examples, okay, for each. Fused genes and patched genes for segment polarity genes. Then they activate, that means pay rule genes activate segment polarity genes. Then the finally homeotic selector genes. This homeotic selector genes is like say approximately, it determines the fate of each segment. Homeotic selector genes, we already knew that regarding basic. There are 3 stages: egg, larva and adult. These are all developing within the egg, then it will come out as larva. Okay, these are within the egg. Okay, and each segment will have their own fate. Homeotic selector genes, it determines the development fate of each segment. But it is not that simple. Actually, it can be organized into 2 gene clusters. Say for instance, antenna pedal and bithorax. Antenna pedal and bithorax. Pedia and bithorax. This antennapedia, for example, say labial. These are different gene cluster 1 and gene cluster 2. Say the labial, say deformed, there are different genes. I told already, different segments have different fates. Say labial gene in labial segments, it will be labial part. Likewise, okay. Deformed, antennapedia. Say bithorax A, abdominal A. I am giving few examples, okay, not entire because lack of time now. Abdominal A and abdominal B. Different things, Say that means homeotic selector genes will be organized into two gene clusters. The antennapedia gene cluster and the bithorax gene cluster. The examples of genes for antennapedia, say the labial, say deformed, say antennapedia. For example, bithorax, say abdominal A, abdominal B. The word it implied. Different segments will develop in different organisms, different organs. The fate will be decided by the homeotic selector. Homeotic selector, this is very important. Say the word abdominal region and labial likewise. So the torso, we already discussed. The torso will determine the anterior and posterior extremities. The anterior extremity is called acron and the posterior telson is the posterior extremities. So the three classes of metal effect genes we have discussed already. Bicoid, this is the most important gene. Then the nanos, the posterior part. Then the torso. In case of zygotic genes, the gap genes, particularly the cripple genes, hunchback genes, and they activate by the zygote metal effect gene, that means proteins, that which are nothing but the transcription factors. Say bicoid is the anterior morphin as well. Then they activate the pale rule gene, that means pale genes like pale and Haley. They in turn they activate the next subsequent genes in a cascading manner. That means the segment polarity genes and ultimately the homeotic genes. It's all about ultimately that leads to proper organogenesis. The first polarity by means of zygotic genes, then the segmentation proper pattern by means of zygotic genes. That means metal effect genes will take care of the polarization, what we call anterior polarity axis, the fundamental block, the fundamental body plan. After the body plan formed by the maternal genes, that means metal effect genes, the segmentation pattern will be taken in the zygotic genes, like gap genes, pale rule genes, like this. And there are different subgenes, there are different genes responsible under different heads that ultimately leads to formation of the proper organogenesis. In the larva, it will come out. But the most significant point is in the pattern formation in Drosophila, the body plan is same in egg, embryo, larva, and the adult. That the structural pattern is same, but the complexity of organogenesis is different. Okay, these are all about different organogenesis. And the most important aspects in examination point of view are gap genes. Why they are considered as gap genes? Because of mutation that leads to formation of gap gene segments. Okay, thanks for watching. You may like and comment, subscribe and share. It is for particularly for the pool and needy students. Okay.